So we have focused most of our Mendelian genetics discussion on pea plants and other just sort of general traits. And pea plants are great because they are a fantastic model organism. They produce a large number of offspring. They have short generation time. They have relatively simple genetics, meaning they have a small number of chromosomes and simple to study traits. And they also they are also easy to grow and easy to maintain. But humans are not model organisms, and so we have to study their inheritance patterns a little bit differently. Now, you can certainly look at two individuals and see that traits are passed on between them to their children, but to actually elucidate how a particular characteristic is actually inherited, whether it's dominant or recessive or some other pattern, we need a different way to study humans, because humans have a small number of offspring. We have a very long generation time. We have a quite complex genome, and we're not very easy to grow. It's a lot of time and effort to grow and maintain humans. Plus, doing controlled genetic breeding studies is frowned upon, understandably so. So we study human inheritance patterns using something called a pedigree. Now, we've seen this before when I drew a little chart for the freckle example. But a pedigree is basically a family tree that shows how individuals are related. So before we get into a big complicated one, let's look at some commonly used symbols. So males are represented by a square, females by a circle. A line in between signifies that they had a child, or they were generally, it indicates that they were married, although it doesn't necessarily mean that they were married, but it does mean that they produced one or more offspring. And so the line down indicates they had offspring together, and then below that we have our second generation. Again, males square, females are circular. If an individual has the affected or has the trait, we tend to call them affected. Now, this does not necessarily mean, it doesn't necessarily have a negative connotation. It can just mean they have blue eyes, but it can also mean they have a particular disease like cystic fibrosis. So the shaded in squares or circles are the affected individuals, and the clear ones are unaffected. If we see a little arrow pointing to an affected individual, this indicates the proband, or the first person that is known to have the disorder. A few other things we might find on a pedigree. This double line in between means that the parents are related, so they might be siblings or cousins. Individuals are often indicated in their birth order. So the farthest on the left will be the oldest, farthest on the right will be the youngest. We can indicate twins. So fraternal twins, these are non-identical twins. We indicate with this line coming down and a split. Identical twins have a line in the middle indicating that they have all of the same genetic information. Fraternal twins are not identical, and so their information is not any more closely related than a regular brother and sister. If we have a lot of individuals in the family and many of them are unaffected, they might be listed with a number inside that just says that there are 4 individuals that were unaffected, and this just is a way to keep everything a little less messy. Here we have another way to indicate the proband, the little p, a deceased individual that may or may not be important to the pedigree. Our book doesn't draw these too often. We can indicate carriers. Now, a carrier is somebody who is basically heterozygous for a particular trait and is unaffected. So we call them a carrier because they can pass on the allele to the next generation, but they don't show it themselves. And then everything is numbered. So Roman numerals on the left indicate the generation number, and then regular numerals indicate the number of the individual number. So we might ask, what is the genotype of individual 2-1? So first individual in generation 2. So let's look at how we might use these pedigrees. So we can determine genotypes from a pedigree, essentially using the same rules as a Punnett square. So there are a few patterns that you might start to pick up on as we look at these. The first is the pattern for a recessive trait. In a recessive trait, 2 unaffected parents produce an affected individual. Now, essentially, this means that those individuals are carriers. So if you see the situation where parents do not have the trait, but the child does, then you are likely dealing with a recessive trait. Now, we can use this information to determine genotypes of our individual. So if we now know that this is recessive, this individual is going to be little a, little a. That means each of his parents had to have at least one little a. Now they were unaffected, so let's fill them in there. And then sister here could be either big A, or big A, little a. So you can sort of see how we can fill in this pedigree just by basically doing a logic problem, inferring based on whatever information we already know. So if we know that this individual is big A, little a, and it's recessive, then we know the parents are heterozygous. And so we can also draw that on the Punnett square. So let's look at an example of a recessive trait. Again, if we look at this pedigree, we will see that these individuals are affected and these parents are unaffected. And that is what I usually look for first when I look at a pedigree. So very often with a pedigree, and probably every time we have a pedigree in this class, I will ask you to determine the mode of inheritance. And once you do that, you will be asked to also determine the genotypes of all individuals. We also see this recessive pattern here, these individuals where you have 2 unaffected parents and affected children. Now, the other thing you might notice is that with recessive traits, they tend to skip a generation or more than one generation. Now, this is not the only way that you can tell a recessive trait, because in a large enough family, you might see it, or if the allele is common enough, you might actually see it in every generation. So you want to look for this unaffected parent and affected children. But if you look at these generations, you can see this generation does not have it, this generation does not, but this one does. And so you can see there that it skips generations. So this pedigree is for something called the ability to taste something called PTC. And this is a little test strip that she's using, and you put it on your tongue and it has sort of a bitter flavor if you can taste it. So tasting PTC is dominant, whereas the inability to taste PTC is recessive, and that trait is indicated by the purple shaded purple shaded individuals. So let's look at the opposite one, at the dominant one. So if you have a dominant trait, it is very likely that an affected parent is going to pass it on to an affected child, and that is because you only need one copy. When we look at dominant traits, I like to look for unaffected children. Let me back up a little bit. All affected children have an affected parent. In other words, you generally don't see skipping in a dominant trait. So let's fill in our genotypes here. Here, if the affected individual is dominant, then we know that each individual has at least one dominant allele. That means everybody who is unaffected is going to be homozygous recessive. So now we can deduce what these guys are. If his daughter is little a, that means that he has to pass on a little a to her. And if his mother is little a, that mother has to pass on the genotype to him. And we can see that in our Punnett square. We have dad, we have mom, and then we have the genotype for the affected boy and the genotype for the unaffected girl. So in this pedigree, we actually can see a dominant pattern. We have every generation expresses the trait. Also, if we look, every child that is affected also has at least one affected parent. Now, the other key thing to the dominant trait is that two affected parents can produce an unaffected individual, and we see that here. So we see two things. We see all affected children have an affected parent, and we see the two affected parents can produce the unaffected offspring. So depending on how you're looking at it, you'll want to look for one of these defining characteristics. Are there unaffected parents with affected children? Are there affected parents who can produce unaffected offspring? So you guys will get some practice in our lab and in our practice problems as well, but I wanted to show you a couple of examples of different pedigrees. So let's look at pedigree A. Let's start here on the left. We have an affected child and an affected mother. That affected mother has an affected father. Each of these affected individuals also has an affected father. So we see all affected children have an affected parent. This indicates that pedigree A is a dominant trait. So let's go through and label some of these genotypes. Each of these individuals is going to have at least one allele that indicates dominance. All of these other individuals are going to be homozygous. I'm not going to fill all of them in, but let's look at these guys. If this son is little a, and mom is little a, dad has to be big A, little a. Now this son here, and likewise the other two daughters, also have to be big A, little a, because mom is recessive. So we can actually fill in all of these genotypes. All the rest are going to be little a. So let's move on to B. Here we see unaffected parents with an affected individual. Again, this is the one that I always look for because it's the easiest to spot. This is therefore our recessive trait. And then the labeling is a little bit easier here. It goes a little quicker. Actually, that's not true. It's easy, but it doesn't go quickly because there's a lot of other individuals here. All these affected individuals are homozygous, and then we can work backwards. So we know if we have two affected individuals and an unaffected child, they both have to be heterozygous. Over here, let's look at, we're just going to do this one individual because all the others in this family are going to be the same. This individual can either be big A, or big A, little a, and we can't determine that without more information. Over here, all of these guys, we can't really determine. We know that they all have to have at least one A. We know that one of these parents had to be heterozygous because they had to pass on to mom, but everybody else we can't really determine. Same thing with these guys down here. These ones are all going to be heterozygous. This individual is more likely to be big A because they have no affected children. If they had an affected child, or if this individual was big A, little a, then we would expect half of our individuals to be affected and half to be recessive, but they actually don't have any of those. So you can sort of deduce that one by the information that you are can figure out from the rest of the pedigree. And then of course these guys over here can't really know for sure what all of them are. You can probably make a good argument for some of them, but we'll go ahead and leave that one there for now. All right, C, again, we have unaffected parents with affected children. So this is another one that is going to be recessive. Now this one is a little bit tricky, and I'm going to point it out just because it's here. This one actually only has affected males, and we will talk about that a little bit later. This is an X-linked recessive, which means that it is found on the X chromosome, and we'll talk about why this pedigree shows only males being affected later. But you can still deduce the recessive part because of the two unaffected parents with an affected child. Our book goes into great detail on discussing how we might use a pedigree. So you can use a pedigree to determine the inheritance pattern, but you can also use it basically in a genetic counseling type situation where you want to determine what the probability that a child will be affected by a particular trait. Now, I'm not going to go through this pedigree because our book actually provides a tutorial. It's on Blackboard in our lecture video section, and it's also on Dashboard under Chapter 5. You can watch that tutorial there, and it shows you how to actually go through and calculate the probability of this individual being affected by the trait. And there are some key things that you have to think about and use our rules of probability to actually walk through this. If you have questions on this, please let me know next time we meet, and I will go through this more specifically. But for now, go ahead and watch that tutorial just like it's part of our lecture videos and see if you can anticipate how they're going to do this problem.